In fact, approximately 40% of you correctly identified this as an osmosis problem, which is actually up from 25% last year, which is really good. But there's still a number of problems, many people mixing up hypo and hypertonic. Less than half correctly identified the blood as being hypotonic compared to the cells. Water flow was from the blood into the cells and caused an increase in cell volume. So the cell is hypertonic to the blood. That statement is equivalent to the blood is hypotonic to the cell. Remember, these are relational words and you have to understand how they're interrelated. Okay. We ended last lecture with pH and buffers, and I'm going to do a brief recap of this concept, or these concepts, and then I'm going to do a demonstration that hopefully helps you understand this in a bit more detail. The pH of a solution is determined by the free hydrogen ion concentration. Acids increase the free hydrogen ion concentration of a solution. If the pH is less than 7, then the solution is termed acidic. Bases decrease free hydrogen ion concentration. If the pH of a solution is greater than 7, then the solution is termed basic. Buffers resist changes in the pH in a solution. So what they do is they donate hydrogen ions in response to an increase in the solution pH. So if there's a change that results in the pH of the solution going up, then the buffer will donate a hydrogen ion. They will accept hydrogen ions in response to a decrease in pH. In the example I talked about last time, I told you that in human blood, the processes that go on in human blood are really sensitive to changes in pH, and the standard pH of human blood is 7.4. To keep it at that level, we have a buffering system called the carbonic acid and bicarbonate buffering system. And I showed you this equation. We have carbonic acid on this side, H2CO3, and this is a hydrogen ion donor. We have bicarbonate, HCO3, it's a hydrogen ion acceptor, and here's the free hydrogen ion. So in this equation, right, we have carbonic acid. If it donates a hydrogen to the solution, then it becomes bicarbonate, and there's a free hydrogen ion in solution. At equilibrium, then, there is no net movement between the concentration of carbonic acid and bicarbonate, and that's at pH 7.4. Okay, at the end of the last lecture, I asked you a question. I said, well, what happens if I change the acidity of the solution? What happens to the buffering system? And I'm going to ask you a similar question this time, but it's sort of flipped on its head. And it's when NaOH, a strong base, is added to the blood, which of the following occurs? Okay, go ahead and click in if you have not. Okay, now before I show you the answer to that question, I'm going to do a demonstration and see if that helps you understand it a little bit better. Okay, so to do this demonstration, I need 3 volunteers from this side of the room and 3 volunteers from this side of the room. So, 1, 2, and people that haven't volunteered before would be great. And way up in the back on the stairs. And then 1 in the red shirt, yep. And then 1 here. And so that's 1, 2, 3, 4, 5. Was there somebody here I said already? You, why don't you come up and then I need, okay. Oh wait, sorry, no, I already have 6. Oh, I'm sorry, next time. Okay, so I have 6, is that right? Wait, no, I only have 4, okay. I saw, okay, please come up, I'm sorry. And then I still need 1 more volunteer. Okay, thank you. Okay, so what we're going to do is, why don't you guys stand back here and look out to, these guys are going to be part of the blood buffering system, and this is going to be the blood, the stage. So what do I have in blood? Water. Well, right, that's the largest component in blood. What else do I have in blood? I have cells. Do I have proteins? Yeah, I have proteins, right? Albumin was one you learned from another question. And you also have the buffering system. Okay. So, let's see. Here we go. Okay, very good. All right. So now we have in our blood, we want our blood to be pH 7.4, and I'm going to tell you each one of these is a free hydrogen ion. And when I place them here, free in the blood, 3 of these are pH is 7.4. Okay, so now we have our buffering system, and we have 2 components, carbonic acid and bicarbonate. So can anybody tell me how we get all our carbonic acids over here and all our bicarbonates over here? Who's carbonic acid? Anybody holding a tennis ball, right? So you should be right here. So I can come over here. So line up this way. Yeah. And you guys are what? Bicarbonates. Why don't you come over here so you're lined up as this, and you face each other. Okay, very good. Okay, so now we're at pH 7.4. We have 3 free hydrogen ions. And at equilibrium, you don't always hold on to that hydrogen, right? You can donate it. So if you let go and sent it over, what would happen? So let it go. Yeah. Very good. Right? So what would happen is we still have 3 carbonic acids, raise your hands, carbonic acids, and 3 bicarbonates. Okay? Raise your hands, bicarbonates, right? So one of our carbonic acids turned into bicarbonate, right? So now I am an agent that comes in, and what am I doing to the pH of the solution? Does it go? So is it getting more acidic or more basic? Acidic. More acidic, okay. And so now what does my blood buffer system do to take care of this? Very good. Is that very good? Okay, now we have kind of a conundrum. Okay, so how many free hydrogen ions are we supposed to have? 3. Very good. Okay. So now we still have 3 free hydrogen ions. And so we're at pH 7.4, and our buffers did that by actually sucking up the extra hydrogen ions. But not all of the buffers, right, had to suck them up because that would take too many out of solution, so we would change the pH. Okay, so very good. So let's reset now. Let's need 3 of these. Okay, good. Okay, now we have some NaOH that is going to come into solution. Here it comes. And then Becca, NaOH, and which one of you, what happens when they come into this solution? Okay, so they had to dissociate, okay, and what binds with hydrogen to make water? Okay, the OH, Becca is OH. Okay, very good, and now what happens to the blood buffering system? Okay. Very good. Okay, so now we have how many carbonic acids left? 2. And we have how many bicarbonates? 4. Okay, so by putting in OH into the solution, OH took one of those hydrogens out, and what happened is, do you want to add one? What happens if you release it? Go ahead, release it. What's going to happen? And what's the blood buffer system going to do? What are the bicarbonates are going to pick them up, right? So you're always going to have, in this situation, a 3 free hydrogen ions, and your buffer system is going to try and take care of it. Now, if I put in 100 tennis balls, what would happen? right? It would become very acidic, and your buffer couldn't take care of it. So the buffer can't always take care of it. But if there are small changes in pH, the buffer takes care of it by releasing free hydrogen ions or binding free hydrogen ions. If they release, then you get more of what? More bicarbonate. And if they actually bind hydrogen ions, then you get more carbonic acid. Okay, very good. Thank you, guys. Put your Okay, now, I'm going to have you answer this question again. Okay, most of you have clicked in. Go ahead and finish if you haven't. Okay, excellent. 79% of you picked the correct answer, which is carbonic acid will release free hydrogen ions. So NaOH, Becca and Ann, they came in, and Becca picked up a free hydrogen ion, combined the OH with the H and made water, and so the carbonic acid released a free hydrogen ion, so it would bring the pH back down to the neutral pH. Okay. Now we're going to answer this question that's actually in your notes, and you're going to answer it as a class card. And it's to take this information that you just learned and apply it in a more complex fashion. So the graph below shows the bicarbonate concentrations in the blood of one normal patient. Draw a graph on your card that indicates if the bicarbonate concentration in the blood of the following three patients is higher, lower, or the same as the normal patient. Bar height should be 1, 2, or 3. So on your card, make a y-axis that says bicarbonate concentration, an x-axis that says normal A, B, and C, and then draw your bar heights for these three patients. And then we'll discuss it. I'll give you about 4 minutes, 3 to 4 minutes to do this. Okay, finish up and pass your cards to the aisles. TAs will come pick them up. And while they're picking them up, we'll go over the problem. Okay. The first one was a patient on a low-carb diet that increases free hydrogen ions in the blood. What does that do to the bicarbonate concentration? So is it going up? So the free hydrogen is high in the blood. It's going to bind to what? Bicarbonate. Okay, and so what's going to happen to the bicarbonate? How many people say decrease? Raise your hands. How many people say increase? Raise your hands. Okay, decrease is correct. And it's because, right, our free hydrogen ions are bound by a bicarbonate. Our bicarbonate then forms what with that, when it sucks up that hydrogen ion? Carbonic acid. So if this question had asked what happens to the carbonic acid concentration, you would have said it increases, right? So you have to be able to go look at what happens to both the bicarbonate and the carbonic acid, because really we're talking about the same molecule that either has a hydrogen or doesn't. Okay, now patient B has swine flu and vomiting decreased hydrogen ions in the blood. What would happen? Increase or decrease in bicarbonate? Increase is correct, and that's because we have a decrease in the amount of hydrogen ions in the blood, so your carbonic acid is going to release free hydrogen ions, and so your amount of bicarbonate is going to go up because all those carbonic acids that donate their hydrogens turn into bicarbonates. Okay, and the final one is patient C has ingested a poison that released OH into the blood. What's going to happen to the bicarbonate concentration? Increase is correct. Again, it will be, right, the OH is going to combine with the free hydrogen ions, make water, take them out of solution, so then the carbonic acid is going to donate a hydrogen to solution, thus making more bicarbonate. Okay. So this is a question that was from the exam 2 years ago. So you have to be able to know how to do this type of manipulation with the OH and the bicarbonate and carbonic acid system. Last time we talked about water. This time we're going to focus on carbon-based compounds, looking at carbon-carbon bonds, which we have already done some of, and carbon bonded to other elements, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. And we're going to be talking about the 4 major macromolecules: carbohydrates, lipids, proteins, and nucleic acids. We'll focus on these 2 today. Carbon atoms are the building blocks of all biological molecules, and they have 4 valence electrons, as shown here in their outer shell. There's 4 electrons, so they can form 4 covalent bonds, either with other carbons or with other atoms. There are 2 major factors contributing to the molecular diversity of carbon compounds. And the first is that there's a large variation in the arrangement of the carbon skeleton. And even here, where we're talking about very small carbon compounds, there are different arrangements. So for example, here in this 4-carbon compound, molecule that has 4 carbons, it can be linear or it can form a T-shape. And these 2 molecules, even though they have exactly the same number of carbons and hydrogens, have very different properties. They can have single bonds between all their carbons, then they're called saturated, right? Or double bonds between some of the carbons, and then they're called unsaturated. They can also form not just linear structures, but ring structures, as shown here. So the actual structure of the skeleton can determine functional properties. All of these are small hydrocarbons, and they contain only hydrogen and carbon bonds. These are nonpolar covalent bonds. So does that mean that these are hydrophobic or hydrophilic molecules? Hydrophobic, right? Because they don't have charges, therefore they're not easily able to interact with the slight positive or negative charges that you have in water. Okay, so these are hydrophobic. Okay, the second major factor that's contributing to the diversity of these compounds are that there are a number of functional groups that can replace hydrogens, and it's the location, type, and number of different functional groups that determine functional properties of the molecules. There are 6 major functional groups that we're going to be talking about in this class, and you're going to need to know the names, the structure, and the function of all of these. Okay, hydroxyl is the first one. It's an OH, which is oxygen bound to a hydrogen. It's shown down here in this molecule. And remember, oxygen has this very electronegative, it's a very electronegative atom. It hogs the electrons from those atoms it's bound to, and so it creates a partial charge, so creates a polar bond, and these polar bonds increase the water solubility because the partial positive and partial negative charges can interact with water. The second group is called the Carbonyl Group. It's a carbon with a double bond to an oxygen. If it's at the end of the molecule, then it is called an aldehyde, and if it's in the middle, it's called a ketone. And this, again, confers very different functional properties on these molecules. And the oxygen, remember, is going to hog the electron, so these also are polar bonds in this molecule when you have the oxygen, and so this increases the water solubility. So if you see groups, functional groups, or molecules with carbonyl functional groups, you know that if they have a lot of them, they will probably be soluble in water. We also have carboxyl, which is the third group. This is an acidic group. We have carbon shown here with a double bond to oxygen and a bond here to OH. This group is able to donate a hydrogen ion to solution, and so it's called an acidic group, because we know when we increase the free hydrogen ion concentration in solution, we make the solution more acidic. Okay, this group can be shown in two different formats. This is called the non-ionized form, and this is the ionized form. So this is the one that is hanging on to its hydrogen, and this is the one that is donated a free hydrogen ion to solution, and it has a negative charge. These, right, if this one then binds back on, then it forms this. You'll see both of these. Both of these are considered acidic groups, right? It's the same group, it's just whether it's donated its hydrogen ion or not. And this is the dominant form seen at pH 7, because at pH 7 in biological, that's a normal, around the normal pH of biological systems. And at this normal pH, pH 7, this will have donated its hydrogen ion to solution, so it's acted like an acid. So if you see this form or that form, you know it's an acidic group. It also has oxygens that are hogging the electrons, so it also is soluble in water. Okay, and then we have the amino group, NH2. This is a base. It's a hydrogen ion acceptor. Here is the nitrogen bound to a carbon, and it has two hydrogens bound to it. This is the ionized form, where it doesn't have the, this is the non-ionized form, rather, and this is the ionized form. So this is the dominant form at pH 7 as well. It has actually attracted a hydrogen, so it's taken on an extra one, so it has an extra charge. So this has a plus charge, and this has a negative charge, right? Because this one's accepted an extra hydrogen, this one has not. Okay, and our fifth group is the sulfhydryl group, SH. This is called a thiol group as well. So it's sulfhydryl or thiol. And this is important in stabilizing protein structure, as we'll see in next lecture, because two sulfurs that come up next to each other can form a covalent bond, and it's the covalent bond between sulfurs that are important in stabilizing 3D structure of proteins. So here is the group, SH, sulfhydryl group, also called a thiol. Okay, and the last group is a phosphate group. It's a phosphate which is attached to 3 oxygens, and that's shown over here. Here's the phosphate with, actually 4 oxygens, 4 oxygens around it. And it's important in transferring energy between organic molecules. We'll see it in the format of ATP quite a bit, and phosphates associated with GTP as well. Both molecules important for cells to do work. Okay, and this also is highly negatively charged, right? It's got released its hydrogen ions to solution, and so this one is also quite soluble in water. Okay, those are the 6 groups. You're going to have to know them and recognize them, and we'll talk about them a lot in the course of the next 5 or 6 lectures. The 4 carbon-based macromolecules that we'll talk about are carbohydrates, proteins, nucleic acids, and lipids. The first 3 are all polymers, meaning they're long molecules built from small similar subunits, or monomers, and they're linked by covalent bonds. In contrast, lipids, they are built from 2 or more different types of smaller subunits. They're still macromolecules, but the subunits that are put together don't look exactly like each other, whereas these guys, each of the monomers that you string together are very similar. Okay. Before we look at the lipids and carbohydrates separately, all of the macromolecules that we're going to talk about are put together by a dehydration reaction, and they're broken down by a hydrolysis reaction. So formation of macromolecules, dehydration. Let's look at that. We have a short polymer here. It is 3 subunits long, and it's now going to be joined to a monomer here. The dehydration reaction involves an enzyme and the removal of hydrogen and 2 hydrogens, one from here and here, and an oxygen, and it forms what? Water, and that's why it's called a dehydration reaction, because it's loss of water. you form a covalent bond between these two subunits. The name of this covalent bond depends on which type of macromolecule we're talking about. And there will be 4 different macromolecules. That means there are 4 different bond types you have to know. Disassembly of these macromolecules occurs by the process of hydrolysis. Again, an enzyme-mediated reaction in the presence of water. Then you can break this bond in between any of these monomers, and you add a hydrogen to one side and an OH group to the other side, and you end up with splitting this 4-subunit polymer into a 3-subunit polymer and a monomer. Okay, so here is a short animation from your book.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking.
Okay, so that's how they're put together. Dehydration reactions, that's how they're put together, and hydrolysis is how they're split apart. Now we'll move on to carbohydrates, which are sugars and their polymers. And the simplest carbohydrate is a monosaccharide, and they can come in either 3, 5, or 6 carbon sugars, and glucose, a 6 carbon sugar, is the most common, which is shown over here. These are all polar molecules in that they have lots of oxygens that are hogging the electrons, so they have partial regions of partial positive and negative charge to interact with water. They are important as a component of fuel in cellular respiration, as we'll discuss in lecture 11. And because they're quite soluble in water, they don't go across the membrane of the cells very well, and that's where cellular respiration occurs, right inside the cell. So these have to be transported across the lipid bilayer. And they're an important carbon source to build other macromolecules. Monosaccharides can occur in linear form, like we just showed you. So here is the linear form, but when they're in solution, they can also form rings, as shown here. So this is the linear form, and then it can form a ring. I don't want you to memorize the numbers of any of these carbons or who's hooked to whom, but I want you to be able to recognize basic features. So for example, if I showed you a 6-carbon ring with a bunch of OH groups on it, I would want you to be able to know that it's going to be reasonably soluble in water because it has these polar groups on it, and I might show you something that looked like this and ask you what type of bond does it form when it comes up to another one that's just like it, and I'll show you in just a second that it will be a glycosidic bond. So you have to understand the basic structure and what the functional groups do in terms of conferring different properties. Okay, a disaccharide is when you have 2 monosaccharides joined by a glycosidic bond. This is a covalent bond, and it's formed by what type of reaction? A dehydration reaction, which is shown here. Here is glucose being hooked up to fructose. An OH group is removed from glucose and H from fructose to form water, and you form a bond that looks like this, and that forms the disaccharide sucrose, and that is a glycosidic linkage. So carbohydrates have glycosidic linkages between, or bonds between their subunits. That's 2 together as a disaccharide. Polysaccharides are when you have polymers of hundreds to thousands of monosaccharides. You can have, well, it's actually anything more than 2. and you can have up to thousands. And they're important for lots of things. For example, structure in plant cells, we have cellulose is a polysaccharide that is important in cell walls in plants. And they're really important for energy storage in the form of starch in plants and glycogen in animals. So I have a short song that highlights the importance of glucose.
Glucose, sugar, sugar, you are my favorite fuel. Corn, such great food. I'm glucose, monosaccharide sugar. You're sweeter than a woman's kiss. 'Cause I need you for glycolysis.
Okay, it goes on and on. But I particularly like that song because I went to camp when I was in 5th grade for the summer for a whole month. That was the first time I was away from home. And we had in my cabin a little turntable in 145, and it was that song, except it was Sugar, Sugar. And we listened to it, I think, at least 1,000 times. So when I heard it in this glucose format, I thought, oh, that's perfect. Okay, moving on to lipids. Remember, they are macromolecules, but they're not polymers. They're hydrophobic because they're dominated by non-polar covalent bonds. And there are 3 forms that you have to know about: fats, phospholipids, and steroids. A fat is a glycerol molecule combined with 3 fatty acid chains. This is done by a dehydration reaction to form a covalent bond that's called an ester bond. So in lipids, it's an ester bond that links the subunits. In carbohydrates, it's a glycosidic bond. Okay, here's glycerol, 3-carbon molecule, OHs on each of these carbons, and it is linked up to fatty acid. This is a long chain of carbons, so it looks like a hydrocarbon tail, right, from a phospholipid. And what group is at this end? There's this, but it's got a double bond O here, so it's a carboxyl, right? It's a C double bond OH. That's why it's called a fatty acid, because it has an acidic group here. This acidic group combines with the OH in a dehydration reaction, and you form an ester linkage. In a fat, you have the glycerol that is attached to 3 fatty acid chains. This forms a fat molecule, and this is really important for energy storage. A gram of fat contains twice as much energy as a gram of carbohydrate. We have phospholipids. Glycerol again, here it is, 3 carbons. To 2 of those carbons, we have fatty acid chains hooked on. One that's saturated and one that's unsaturated. And at the head region, we have a group, what's that group called? Phosphate group, exactly. And so this group is charged, and so that actually makes this part of the molecule hydrophilic or hydrophobic? Hydrophilic. That's why the head of the phospholipid is hydrophilic, because it has this phosphate group up in this region. Okay, so we have a hydrophilic region of the head because of the functional groups, and hydrophobic regions in the tails because they're dominated by carbon-hydrogen non-polar covalent bonds. Okay, phospholipids we know are an important component in the membrane, and if you just throw them in water, they have interesting behaviors that are associated with the location of these functional groups and how they confer hydrophilic and hydrophobic properties. So if you put them in water, sometimes they form these structures which are called micelles, with the heads all pointing out and the tails in. So that's increasing the probability that the heads are associated with water and the tails in. And so that's it will form these structures spontaneously based on these charges. They will also form these structures which are called lipisomes, which basically look just like a lipid bilayer. The head of the ones on the outside are, this is facing the water and the tail's in, and here the heads are in facing the water in the middle, and the tails are in the bilayer type region. Okay, and finally they can also form a phospholipid bilayer at the interface of the solution. That's a little bit less common. Okay, and finally we have our third class of lipids, which is steroids. And these are carbon skeletons with 4 rings and varying functional groups. So here are the 4 rings that are attached to each other, and then a number of different functional groups. And the one shown here is called cholesterol, and you know that's an important component of the membrane. In addition, cholesterol is the precursor of all other steroids, including testosterone and estrogen. So from cholesterol, then you can make these other steroids. Okay, so this has pretty hydrophobic region. Is there any region of this that might be hydrophilic? Right? It does have an OH group down here, but in fact, it isn't very hydrophilic because it's the relative abundance of these non-polar covalent bonds to the polar one. So here, it's dominated by non-polar covalent bonds, and there's one little polar bond down here. So it's quite lipid soluble, not very soluble in water. Okay, so I'm going to give you a few minutes to work together with your group to go through and actually now take some of this information I've just gone through with you and apply it to see if you can remember what each of these functional groups is called, if they're charged, polar or nonpolar, and do they confer hydrophilic or hydrophobic properties on the molecule. And then we'll go over it together. Okay, even if you haven't finished, let's go over this together. And for these, I'm just going to ask for shout-outs as a group. And so we're going to go through each one of these. So functional group A is shown here. What's its name? Amino, very good. Is it charged, polar, or non-polar? Charged. It's charged because it has a positive charge. It's actually fully gained an extra hydrogen. So we have, is it hydrophilic or hydrophobic? Hydrophilic, exactly. Okay, B is what? Hydroxyl, very good. Is it charged, polar, or nonpolar? Polar, that's right, because it has oxygen and is creating a partial positive and partial negative charge. Hydrophilic or hydrophobic? Philic is correct. Okay, C is a carbonyl group. Charged, polar, or nonpolar? Polar, that's exactly right, because it has oxygen that is hogging electrons but not actually taking them. Okay, phobic or philic? Philic, very good. It was pointed out to me in the first lecture that this is spelled incorrectly, and it is. This is a methyl group, so it's this carbon with 3 hydrogens. I didn't go over that in the previous 6 groups, but you do have to know what this group is called as well. Is it charged, polar, or nonpolar? Non-polar, because it's got only carbon-hydrogen bonds, which are non-polar covalent bonds. Okay, good. And is it hydrophilic or hydrophobic? Hydrophobic, very good. Okay, E. Carboxyl, very good. Charged, polar, non-polar? Charged, exactly, because it has a negative. And philic or phobic? Philic, very good. Almost done. We have F. Phosphate group, good. Charged? Yep. Hydrophobic or hydrophilic? Hydrophilic. Okay, and our last is sulfhydryl, which is also a thiol group. It is polar. I didn't tell you that, but it does actually have a little bit of, it has a partial charge. And therefore, is it hydrophilic or hydrophobic? Hydrophilic, very good. Okay, and for the last thing, we have a question. Vitamins are characterized as being either water-soluble, hydrophilic, or fat-soluble, hydrophobic. Based on analysis of the functional groups, which of the vitamins shown below is most likely to be in if you haven't. It's getting a bit late.